Hello there. As you can tell from the title, this is a talk about immunology, which is a defense of the body. And I must stress it's aimed at optometrists, particularly optometrists doing independent prescribing. It was originally written to not have voiceover, so there's an awful lot of words in here, but I guess that might help you if you want to go back and read it again. It's also coupled with another talk I've done on allergy, and it's best to watch this one first before you move on to the allergy, because my point is you can't understand allergy really if you don't understand inflammation and immunology. It's important as independent prescribers to understand basic general immunology, the innate system versus adaptive, and why in fact we have two prongs to it in the first place. The innate system includes barriers and it's the first response. Then there's the acquired system that kicks in later, but also the refinements, cult, and also immune privileged sites, which I think are important to at least understand because there is real impact with the eye. Another point to understanding immunology is so that we can actually physically understand published papers. We'll be reading these papers and they will assume we know. They won't explain these principles, they'll just state them. And also it helps us obviously explain to patients, advising them on different things, drug therapies, why we've done certain things, the outcome, what's going to happen, and post-surgical implications as well, so that we can actually explain things. And it is, as clinicians, it's critical that we understand rather than simply learn what we're clinically seeing. Papillae, chemosis, follicles, allergy, toxicity, what do they all represent so that we can treat them better and understand them? And we need to be able to treat conditions confidently and logically. And also then we are then able also to understand allergy. And as I've already stated, to my mind, you cannot understand allergy without understanding inflammation first. Now here's an example. This is a clinical management guideline on GPC, and it states that it's a multifactorial etiology. It's partially type 1, immediate hypersensitivity. It can also be type 4, mediated by T cells, and also it can be elicited by trauma. Now, they don't explain those terms, and why should they? They're expecting you to understand these. Now, for an exam, I guess you could just learn it off by rote and fill in the exam paper, but it's much better to understand the principles behind it. And so many of these things that we'll read just make this absolute assumption that we understand what we're doing. So an overview of the immune system, it's simply a whole gamut of things, protein, cells, organs that defend the host, and it has to recognize self from non-self. A pathogen is an infectious organism causing a disease. A commensal organism is still actually an infectious organism, but it's beneficial to the host, such as our gut bacteria, which of course we couldn't live without. What's the goal of the immune system? Well, it's first of all is simply to neutralize an immediate threat. So that might be the toxins. It's not necessarily there to destroy that thing. It's just we've got to neutralize it first. Then, of course, it comes as destruction or elimination of the pathogen if it's possible. So there's no potential to cause further disease later on, but it's not always possible to do that. And there's mechanisms built in to try to control that possibility. So it must try to eliminate the pathogen first without collateral damage to the host. That's the important bit, and that's tricky. It must recognize self from non-self. Now, of course, under current times, this is 2020, that's an interesting thing because, for instance, drinking Lysol or Domestos will certainly kill the bug, the pathogen, but of course it will also destroy host cell tissues and indeed the host themselves. So, it's an important principle, if a little current at the moment. And the thing about pathogens is it really does test the defense system, because there's all sorts out there. They're different shapes, they're different sizes, hugely different sizes. Some have different life cycles, multiplication rates, and so on, and how they create the pathogenic mechanism as well, and then evasion strategies. You know, some live inside cells, some have waxy coats, some mimic the host. So the immune system has to deal with a humongous amount of things, and still try to save the host from damage itself. So the primary purpose is to protect the host. Okay, we've said that. The first stage is simply barriers. So with the general barriers, we obviously have our very tough keratinized skin. We have all the little organisms that live on us and in us. We have chemicals, lysozyme, and so on. We do have these barriers like the blood-brain barrier, which are really important, which we'll come on to later. We have cilia in our nose and throat, mucosal membranes, goblet cells throughout our body, not just in the eye, the pH in the stomach, and so on. And we have ocular equivalents. We have all our mechanical barriers, the bony orbit, eyelashes, the sebaceous glands, mucosal membranes with our goblet cells in. We have the tears which flush the pH, they prevent adhesion and dilute concentrations and so on. And of course we have chemicals in our tears, as we all know, lysozyme and lactoferrin and so on. So there's an equivalent with the eye. Now these two I will go on to a little bit more later. The hard barriers, the example is the blood-brain barrier, but also ocular immune privilege, which I've mentioned earlier, which is really, really important with the eye. Of course, our barriers are pretty good at protecting us, but if something does penetrate those barriers, then we have to kick in the inflammatory response. And first of all, we have to simply be aware of the infection. There's no point in infection getting in, it's not actually being aware that it's even there. And then once we are made aware of that, the innate immune response, inflammation that is, kicks in. And as we all know, it's immediate, it has no memory, and it's non-specific. Now I know we all know that, we've all learnt that at uni. Do we understand why and so on, but we'll come on to that. The purpose of inflammation is to supply an effective initial response to the invasion, first of all. It's got to recognize that we've been invaded and then get it. But then further that has to recruit reinforcements from the bloodstream to aid destruction and removal of the pathogens and other dead cells. So it does that with vasodilation, activity of endothelium and so on. Now the first cell really that we get is what we call a tissue macrophage. Now by the very name, it's in the tissue already. It's no point it being miles away and comes in. It has to be there. It eliminates the pathogen and then it cleans damaged tissue, dead cells from the area, and it triggers the complement system, which we'll explain a little bit later. And it does it by phagocytosis, so it attaches, ingests and kills and degrades the bugs. And it is really important that it's present for an immediate response. Like I've already said, there's no point in being in your liver. And it recognizes cell surface receptors as foreign and gets them. But usually it is relatively unlikely necessarily to eliminate all pathogens. And so it'll send out chemical messages as well to recruit further help. So it activates other things, communication cytokines, proteins, in the microenvironment and so on. And then it has also direct action chemicals like prostaglandins and so on. So it's doing all this at the same time as other things. Now also in the tissue we have mast cells. And these are distributed throughout the body at potential sites of invasion. They're preloaded, that's really important. So they don't have to manufacture stuff, they're already preloaded, ready to attack. Hence immediate. Histamine, heparin, and so on. So while a tissue macrophage's main role is to phagocytose foreign particles, the mast cell's immediate response is to discharge lytic chemicals to kill. But both also produce messenger chemicals, cytokines, to enlist blood-borne reinforcements. So once triggered, the mast cells do then start to synthesize new stuff, prostaglandins, leukotrienes and so on. And this magnifies and sustains a response, again via more vasodilation, increased vascular permeability and so on. So initially it's got some stuff that's preloaded ready to go, but then once triggered, it starts producing more things as well to magnify the response. But there's a lot more to the innate immune system inflammation than just macrophages and mast cells. We have lots. There's the interferons, which can inhibit viral replication. There's natural killer cells, which their very name tells you what they do. They just take things out. Tumor cells, viruses, bacteria, protozoa. If it's foreign, they'll kill it. Then there's a thing called the complement system. Now, this is a series of proteins and enzymes, and they cause a cascade. And once triggered, they just start. They just roll on. One triggers the next, triggers the next, triggers the next, until they achieve what they're supposed to do. And C3A and C5A cause a degranulation of the mast cells. Now, is it important that we know that? My point about immunology for us optometrists is we do need to know the principles, but the very subtle nuances of what triggers what where, I think, is not necessary for what we need. But anyway, there's also the clotting system, which the clots limit entry of pathogens, and also they have stuff that's chemotactic for phagocytes. So in other words, they attract in other things. So all very important things to know about as part of this inflammation that we're witnessing all the time. So the following slide is an attempt, and I do mean attempt, to graphically illustrate the inflammatory response. And it's not to scale, obviously. So there we are. You can see the blood vessels at the bottom. We've got our tissue macrophage and our mast cells there already. They're already in the surface of the tissue. And in comes some bugs. And step number one, the macrophage will phagocytose, and hopefully phagocytose all of them, and that's the end of the problem. But it's possibly unlikely that that will happen. So then it starts sending out messengers to the blood supply to start drawing in reinforcements from the blood. But the mast cells also trigger their lytic enzymes, and they can blow away some more of them. And again, hopefully that takes them all out. But then the blood vessels start opening, and then we get blood-borne things coming in. More phagocytes and granulocytes that start spitting out more things and so on. And as that goes on and on and on, so we've got eosinophils, basophils, neutrophils. Neutrophils are another phagocyte, only much, much smaller, but they come in from the bloodstream. So this is all spiraling all the time. And that's why inflammation is hot, it's swollen, because we've got all this fluid being pumped into the area that we're trying to control an infection of. And another point I want to make is the innate, I think you probably get this from what I've already said, but the innate immune response is the inflammatory response. They're exactly the same thing. So don't get confused by that. Don't think we're talking about different things. They are exactly the same thing. And no matter how that's triggered, whether it's triggered by something becoming infected or simply trauma, as I often point out to people, if I rub my skin lightly long enough, I will generate an inflammatory response. So a suture constantly rubbing on an upper lid will cause an inflammatory response. It's not infected, but that's the inflammatory response, and that is the innate response. And another thing I'd like to point out, the papillae, well, this is in the CMG. The papillae is hyperplasia of epithelium, and they contain a vascular core. And it is a reflection of the non-specific innate immune system. So sometimes you might hear lecturers say it's a non-specific response, and that's why, because it's only part of the innate system. It hasn't been designed to attack one particular bug. It's there to just defend us. And chemosis is pretty much the same thing. It might look different, but it's pretty much the same thing. But they're different representations of the non-specific inflammation. And papillae form where there's a firm attachment of conjunctiva to underlying tissue. So the tarsal zone but also the limbus. In very bad situations, we'll have limbal papillae. Chemosis tends to occur under the bulbar conjunctiva as there's no firm adhesion, and it just erupts out and balloons out with fluid from the inflammation. So papillae tend to be fairly structural, and I think they tend to happen when it's more chronic as well. If you've got an acute allergic reaction, papillae haven't got really time to develop anyway, but you will get a blowout of chemosis. We'll never be taught that at uni, I don't think, but they are actually just different reflections of the innate immune system. So a quick summary of the innate system. We've got the barrier functions, both physical and chemical, and they are technically considered to be part of the innate system. So it's called innate or natural because it exists prior to invasion. So it simply recognizes self from non-self, and therefore it's non-specific. It has no memory. Every invasion is treated the same. But because the system pre-exists, the response to tissue damage and infection is immediate once recognized. So that's why it's immediate. Really, the defense of our body is a war. The bacteria and the pathogens just want to live happy lives. They don't want to be killed by us, but at the same time, we don't want them there. So it's a constant evolutionary process of how things develop. So some pathogens have worked out ways of evading the innate system. Some of them hide inside our own cells, like viruses and chlamydia, incidentally. Some have waxy coatings, like streptococcus. Some are just huge. Some bacteria are so big they just bulldoze right through. Now, obviously, that goes for parasites as well, but there are actually some bacteria that are like that as well. And sometimes it's just too damn chronic. It just keeps on going. So if the innate system does not succeed, we then need a more specific mechanism. And that is where the acquired immune system is triggered. So the acquired immune response is delayed, because obviously it's not there to start with. It has to be manufactured. It has memory, which we'll come on to later, and it's also very, very specific. So we're creating a particular mechanism that's manufactured solely to defend our body against one specific pathogen. Now, it's only delayed the first time. Because it has a memory and it's specific, once it's primed, it is instant. Right? So we have a memory, we store loads of stuff in plasma cells once it's created. So you must always remember it's only delayed the first time. Now this statement down the bottom here, I'll explain that in the allergy lecture, but this chap, Von P, I call him because I can't say his name, he actually coined a lot of terms we use for allergy and in fact immunology. And he used the term hypersensitive, not to represent a bad thing actually. He coined that term to say that once our acquired immune system is primed, we are then hypersensitive for that specific pathogen. So I always think of people as hypersensitive in more or less a good sense, but in allergy it can be a bad sense. So why the delay? It is a perfectly sensible question to think, well, why can't we be hypersensitized already? Well, it's just the numbers game. It's just the numbers are too big. Someone did the maths on this. To have antibody specific for every antigenic epitope possible would take 10 to the 11 different antibody specificities. And to carry enough, we would need to have 5 tons of antibody in our bodies at all times, which is patently impossible. Now, evolutionarily speaking, since we'll never encounter the vast majority of bugs, the acquired immunity ensures that we maintain immunity to our local bugs. So we get infected once, we then become immune to that. That bug's going to hang around. We may get infected again, but we've already got the antibodies in our body. So instead of having a delayed response, we have an immediate response. But of course, when a bug either evolves differently, like MRSA, or is a new bug introduced to the area, we don't have any immunity to that. We've never encountered it before, so we have to start from scratch. And COVID-19 being, the corking example. It was introduced, and so when it first came here, not one person had any level of immunity for that bug. Because it was a virus, it evaded the innate immune system by living intracellularly, and so we had to delay while we manufactured antibody specifically to come up with a mechanism to control that particular virus. And of course that takes time. And some people's immune systems aren't as prompt or aren't as good, whereas other people's are better. So some people struggle more with it, while other people actually just have quite a minor condition. So there are actually 3 phases to the acquired response, and each one is dealing with a tougher situation, if you will. And the cells of the specific immune system are lymphocytes. That's white blood cells. Now the first role is the generation of antibody, and antibodies, the B lymphocytes produce glycoproteins called antibodies, and also incidentally antibody and immunoglobulin are the same thing, right? So if you read a paper that's used in term antibody or immunoglobulin, they mean the same thing. I tend to say antibody because I can say it better, but Ig, so for instance with hay fever, which is a reflection of this phase, and it'll say hay fever is IgE mediated response. So it's this particular prong of the acquired immune system that's been sort of diverted in an adverse way. And so much about allergy is a reflection, well, all of allergy, if you will, is a reflection of some process here. Secondly, we have the antibody-dependent cell-mediated cytotoxicity. This is very, very important for viruses, CD8 cytotoxic T cells, and they're aimed at intracellular organisms. Now, we'll talk about this a little bit more. Again, do we need to know it? Do we need to know all little nuances of it? I often have to go back to the books to reacquaint myself with it before I do a talk. But understanding it, so the clinical management guidelines might say it's a cytotoxic T cell reaction. So it's important to understand the process. And then the final one is delayed type hypersensitivity. This is a magnification process which recruits it a lot more. So we're talking about everything else has kind of failed at this point, and it's really bringing in the big guns to try to really bring in the hypersensitivity so we can really combat the particular bacteria, or particular bug, I should say. And also there's a reflection there in, for instance, a type 4 allergy is more or less a reflection of this delayed type hypersensitivity. So everything we see in allergy is a reflection of the needed and essential acquired immune system. So the antibody response, this is the nicest one really. Once primed, we produce these antibodies specific to the pathogen. And these, obviously, we've already said they have to be manufactured post initial infection. But once produced, volumes are stored in these plasma cells. So once we're carrying lots of plasma cells just loaded down with a very, very specific antibody. And on reinfection, the response is rapid and specific. There are 5 antibodies. There's a pentamer, that's IgM. We'll get onto that. IgA we should know because that's in our tiers a lot, and IgD, IgE, and IgG. So the general antibody structure, they're Y-shaped, but they have a variable region. Now the other bit, the constant region, is the same. So if every IgA will have exactly the same constant region. It's the variable region that varies and gives it its specific specificity for one antigen. And they have to be developed. But the rest of the structure is all the same. The antibody doesn't bind to an entire antigen, so it can't bind to a whole thing. but it binds to an amino acid sequence matching the variable region called an antigenic epitope. But the thing is, how do we get something that specific? How do the variable regions with high affinity for a specific antigenic epitope, how are they created? Because they don't just pop out, they have to be made post-infection. So on first contact with a new novel, for that body anyway, pathogen, B cells will express IgM on their cell surfaces. Now the role of IgM is to differentiate into another specific antibody with high affinity for that specific antigen. So IgM doesn't actually have an antimicrobial action itself. It is simply the precursor of everything else. So let's go through it. We have this thing, clonal selection. So for instance, we've got these things floating around, and say 1 in 100,000 B cells, by complete chance, have IgMs on their cell surface with affinity for that particular antigen. The other ones just float on by, right? But this one just happens to have affinity for it. And then they go through, once triggered, again it's another cascade, they have clonal expansion. So those B cells undergo extensive cell division, cloning themselves rapidly and increasing the number of B cells with antigenic specificity. Then they do the class switch. The IgM then switches class to a specific one, IgA, IgG, or IgE. And then we go through affinity maturation. And okay, you can look these up in books and read all the fine details, but again, the principle is it has to go through these processes to spit out what we want. And that antibody continues to mature and develop stronger affinity via somatic mutation. So this is a completely random process. And then finally, the B cells differentiate into plasma cells, which start secreting the specific IgA, IgG, or IgE that's been designed. So they have mass of this stuff. They're just floating around with loads of it. And then we create memory cells. And they keep that specific IgA, IgG, or IgE on their cell surfaces ready. And these are very long living. So again, if a bug comes back in, we're already ready for it. So we might only have a very, very small number of IgMs with a specificity for that particular antigen. But by the time we've gone through that process, we're spitting out IgA, say, loads and loads and loads of stuff, secreting masses of Ig specifically for that particular antigen. Now, the acquired immune system wants to produce enough antibody to control that invasion as quickly as possible. So it uses different techniques to try to speed the process up. And one of them is affinity maturation, which we mentioned earlier. And in this, the antibody continues to mature and develop stronger affinity via somatic mutations, but it's mutations, so it's completely by chance. So for instance, you'll have, say, a particular IgA that may have affinity for the pathogen, but it hasn't got high affinity for it. So rather than discarding it completely and just picking up the ones that already have high affinity, that's going to take too long. So through this random mutation, some of them become higher affinity variable regions, and these compete favorably and they continue to be refined. Whereas B cells not stimulated by the antigen, they just die off. So we're sort of funneling the system down to produce a load of antibody with high affinity for that particular pathogen. But here's the rub. It's totally random. And therefore, you could have a random mutation that created antibody with affinity for the host. And that's where we enter T lymphocytes. So the T stands for thymus-derived. helper cells are so called because they were found to be really, really vital to the process, but they didn't actually have any antimicrobial activity themselves, but they are essential to help other ones do it. And the beautiful thing about this is they recognize pathogen in a totally different way to B cells. And this is really, really critical. So it's like a fail-safe system. It's for the system to work, it has to be recognized by two different processes at the same time. And so it always amazes me that we have any allergy at all, because these systems are so well evolved. So all the processes we've talked about before, generation of antibody, the production of CD8 cytotoxic T cells, and the delay, they all depend on T cells. And as we've already said, while the T cells do not actively destroy pathogens, they are vital to initiate all the other processes. Okay, let's say it one more time. No acquired immune response can happen without T cells. But T cells cannot, if you remember, B cells just recognize free pathogen. T cells cannot. They can only recognize antigen presented to them by antigen presenting cells. And these aren't always specific cells coming around. Macrophages are an example of an APC. And they express proteins on their cell surfaces called major histocompatibility complexes. And CD4 T cells only become these T helper cells once stimulated by pathogen presented to them on these antigen presenting cells. So once a naive CD4 T cell has been stimulated via recognition of antigen presented on a class 2 major histocompatibility complex on an antigen presenting cell such as a macrophage or a phagocyte, they then become T helper cells. So initially they're just CD4 T cells. They become T helper cells, and that allows then the proliferation of B cells and the differentiation to plasma cells. So those cells cannot do it without T helper cells. They'll also differentiate into what's called CD8 T cells, which become CD8 cytotoxic T cells, and these are really important for viruses. And they also promote recruitment of cells for the delayed type hypersensitivity. So all three sections of the acquired immune system. For us, I think we should just think that there are basically two. There's a class 1 MHC and there's class 2 MHC. There are more, but they're the ones that we really need to concentrate on. Why do we have them, and why do we have CD4 and CD8 T cells? Well, it's partially because they're guiding them towards a different thing. One is directing the immune system toward an antibody response, and the other one is directing them to what's called a cell-mediated response, and I will explain that. An exogenous antigen, like a bacteria, so it lives outside the cell, it's processed by MHC2, and it's recognized by CD4 T cells. Endogenous antigens, like a virus, which lives inside the cell, is processed by MHC1 and then recognized by CD8 cytotoxic T cells. So the complexities of this acquired immune system with its three prongs has to be instigated somewhere. It doesn't just appear. And it's the lymph nodes. And the lymph nodes are the factories where all this complexity happens. And inside the lymph nodes are germinal centers called follicles. And I think I want you to latch onto that term as optometrists because it'll come up in the next slide. And these lymph glands are the germinal centers where pathogen is recognized and the production of antibody, CD8 cytotoxic T cells, and the delayed type hypersensitivity are instigated. But imagine the response would be quicker if a pathogen did not have to be carried all the way to a lymph node. So at this point, all that previous theoretical stuff becomes clinically significant, and that's why the principles are important. So I want to introduce the concept of MALT, mucosal associated lymphoid tissue. So what the body does is at sites of exposure, like mucous membranes, gut, urinary tract, and of course the conjunctiva, are lined with lymphoid tissue. So rather than going all the way to the lymph nodes, they get taken out with this lymphoid tissue. These are follicles with germinal centers and dendritic cells for antigen presenting cells for stimulation of T cells, as well as the antibodies themselves. And in the eye, we simply decided to call it COLT. It's still MALT, but it's conjunctival associated lymphoid tissue. And indeed, follicles are COLT. And the clinical management guidelines define follicles as hyperplasia of lymphoid tissue. And this is hence the clear appearance. They're fundamentally, just think of them, well they are, don't just think of them, they are. They're mini lymph nodes. And therefore, when they are hyperplased, they are representing a very specific response to infection. You know, at uni we're taught to say, well, this sort of infection will have a follicular reaction, this sort of infection will have a papillary reaction, and that's okay if you learn it by rote and you can remember them all, including all the exclusions. But the bottom line is simply to understand what they represent in an immune response, and it clearly gives us more information than simply learning things by rote. And virtually all plasma cells produced in the MALT, and by default therefore COLT, is secretory IgA, as we all know we have levels of in the tears. Just briefly, before we go on to these next 5 slides, just another comment about papillae and follicles. I think when we're at uni, we're sort of led to believe that they are quite distinct animals and you can't have one, or if you've got one, you haven't got the other. Well, of course, that's just illogical. You know, if a really virulent bacteria has bulldozed through the innate system, it's obviously stimulated the innate system first, so you may have a papillary response. But then if the innate system can't control that bacteria, then the acquired immune system can be kicked in, and that will obviously produce a follicular response. So there's absolutely nothing stopping papillae and follicles coexisting in the same infected eye. So just something to always keep in mind, because compartmentalizing things in nature doesn't actually always work. But anyway, onto antibodies for a moment. We've got these Y-shaped proteins that are developed, and you think to yourself, well, okay, how do they do the job? Right? They've got to destroy or neutralize pathogens, so how the heck do they do it? Well, there's 5 ways actually. The first one is neutralization. So simply binding to something, they can do it, like toxins, or if they bind to a virus or a bacteria, their antibody can actually inhibit its action. So toxins, for instance, with tetanus, the toxin is the entire disease process. So the antibody can bind to the toxins and simply stop them performing. Now with viruses, of course, we realize that antibodies can't neutralize an intracellular virus. But viruses do have to bind to cell surface of host molecules to gain entry. So antibodies, by binding to the viral receptor molecules, they can actually prevent the virus getting into the cell. But also, once a virus gets into a cell, it propagates, breeds up, and then effectively it lyses, so it erupts out of the cell and then starts spreading elsewhere and infecting other cells. Now, once an extracellular virus is out there, again, antibodies can very often bind to that extracellular virus, ensuring that it's neutralized before it can gain entry to another host cell. And same goes with bacteria, really. Apart from also binding to the toxins, if that's appropriate, they can also inhibit bacterial metabolism by binding and inhibiting nutrition or nutrient transport. So neutralization can work in a host of ways. The next one is agglutination. And basically, as the name implies, it just basically clumps everything together and stops the whole thing functioning. Okay, it says it's multivalent, so it can bind to multiple microparticles, but basically it just clumps the whole thing together and stops them working. But also, relatively large complexes are also more easily phagocytosed by our normal innate macrophages. There's a trick to this though. If they become too big, the complexes may not be cleared. And so we can use a different sort of format to get rid of these big complexes. Number 3 is opsonization. So this term comes from the Greek word to prepare for the table. So it's trying to make a microbe more appetizing and ensure that macrophages will destroy it. Now, this process exists in the innate immune system as well. It's been going on a lot more, but sometimes some bugs can outwit the innate system, as you know, and if you've got a very specific antibody for that bacteria, it can make the opsonization process and the recognition by phagocytes of the innate immune system much better. So it speeds the process up. And then there's activation of complement. Now, we've mentioned this earlier on, and it's part of the innate immune system. It's simply a cascade of chemical molecules, and once triggered, it just erupts and keeps on going, triggering one after another down different cascades. And it will attack bacteria or pathogens anyway in a variety of ways. Some of the chemicals can simply lyse pathogens, so they get on there and just erupt. Then there's also others that help with opsonization as well. So again, attracting monocytes and macrophages to come and destroy the pathogen. Chemotaxis recruits cells from the innate system to magnify the inflammatory response. So again, here's an acquired immune system triggering an innate response or speeding up an innate response. So it's a very, you can see it's a very, very complicated system. It's all interwoven. It's not, oh my goodness, one section's finished, we'll start another. It's all muddled up and interwoven. Then there's inflammatory mediators. So they can activate mast cells, eosinophils to degranulate, and so on, neutrophils. So again, it's just, again, the innate immune system, but being triggered much more effectively by an acquired system. And then there's clearance of immune complexes. So these antibody-antigen complexes can get trapped in blood vessels, and they themselves cause inflammatory disease, which is one of the great downers. But some of the complement system help limit the size of the complex lattice, and they can prevent deposition in vessels. And finally, there's what's called antibody-dependent cell-mediated cytotoxicity, ADCC for short. So this is at a much higher level. This is when things are getting a bit tough, actually. Host cells infected by intracellular viruses, bacteria, or parasites may express antigen from the pathogen on the cell surface. This is recognized by antibody, and the host cell is killed by natural killer cells and macrophages. So again, all right, we mentioned those things in passing before, but suddenly they're being triggered again by the acquired system. Killing is extracellular. That is to say, they're not phagocytosed. Extracellular killing has been noted for big worms and so on. And IgG and IgE promote this process. And a comment in allergy, IgE pretty much now, because we've evolved, doesn't seem to have any real purpose in our bodies apart from causing us distress by causing allergic type reactions, anaphylactic type reactions. Well, one of the arguments is that one of its main reasons is to take on these much bigger things, big worms and so on, and helping to destroy them via this process ADCC. So while antibody can do a lot if the pathogen is extracellular, and that includes viruses so long as they're extracellular, they cannot eliminate intracellular pathogens. For the eye, we think of viruses, simplex and zoster, bacteria, chlamydia, but also fungi and protozoa, although I can't think of any examples for the eye. But they're the main ones that we'd be dealing with. And what happens here is, because it's inside the cell, we've actually got to destroy the cell effectively. We've got to lyse the actual host cell. So there's some level of collateral damage with this. And this is why there's a different class. Basically, an infected host cell will present class 1 MHCs on its cell surface. But unlike antibody, on first encounter with a specific class 1, the naive CD8 T cell cannot act immediately, but rather it has to be stimulated then to become a CD8 cytotoxic T cell. So CD8 T cells recognize antigen presented on these class 1, and they're on an infected host cell. So first presentation, no kill, like developing antibody in the first place and so on. On first presentation, we don't do anything. We have to develop the system. The CD8 T cell is activated with the help of CD4 T cells, and the CD8 T cell specific for the class 1 MHC will proliferate. And these proliferating cells differentiate into CD4 T cells, and they're ready to kill any host cell presenting with that MHC on its surface. So it's not actually killing the pathogen, it's killing the cell that holds the pathogen. And this becomes important later on because we want to do as little damage as possible. And sometimes it's not in the host's best interest to go killing its own cells. So anyway, cell-mediated cytotoxicity, basically they kill in a couple of ways. They exocytosis, they have perforin, which protein that punctures host cells causing cell lysis. And then there's Fas molecule, which, okay, do we understand it? Do I understand it? I don't know. Is a death molecule on some cells triggered by CD8. So, okay, do we need to know all that? Don't think so. Just understand why in journals they'll say, well, it's a T cell mediated mechanism. And CD8 cytotoxic T cells can be recycled. So after the lethal hit, it can detach from the dying host cell and can kill other target cells, which is obviously quite natty. But sometimes, you know, as good as this system is, it's huge sometimes. But even then, it's not always enough to eliminate intracellular pathogens. And that's when we trigger up the delayed type hypersensitivity. So, all right, all else has failed. The pathogen is still there and it's still causing disease. So at that point, we basically bring up the delayed type hypersensitivity, which is in simple terms, really, the way I think of it, just a huge magnification loop of the whole process. So we're getting pretty destructive at this point, incidentally, of our own host tissue. It's one of the great examples of this is a pathogen that decides to live inside the macrophages. And obviously that makes sense. That's tricky because when you think about it, everything we talked about with the innate immune system, and then so much that we've dealt with in the acquired immune system, is trying to ensure that macrophages can phagocytose and destroy pathogens. So of course, if the pathogen is actually living inside a macrophage, that all becomes fairly tricky. So again, it's a magnification loop. It's trying to, basically, very simply down the left there, it recruits monocytes to the site of the infection. It retains those monocytes at the site of infection, and it activates them to kill intracellular pathogens. Now, if you like, you can stop the slide there and read the 7 points down the right. Will you remember them after those points afterwards? I think it's important to read them, but have a go at that. But basically, you get the point. It's a big process, a huge magnification loop. So once these resting macrophages are stimulated by cytokines, they simply get activated and are more effective phagocytes and produce lytic enzymes for more effective killing. And once they're recruited to the DTH site, they stimulate each other, providing this, again, as I've already said, this amplification loop, increasing the magnitude of the delayed type hypersensitivity response. So, you know, again, this is getting huge now, and there is a load of collateral damage. So as we said in that last slide, you know, the delayed type hypersensitivity is really quite an aggressive process, and it does cause a lot of injury to the actual host. And you've got to remember that with the immune system, it's got two things to do. It's got to eliminate the pathogen if it can. Certainly it has to do that. That's its goal. But it also has to do least harm to the host. There's no point destroying the bacteria or whatever it is if you then destroy the host. So antibody responses are generally the least damaging. You know, especially neutralization. If an antibody can neutralize an extracellular toxin, there's effectively no risk to the host whatsoever. CD8 cytotoxic responses, of course, are directed at infected host cells. So there has to be host damage. You are killing a host cell to destroy the intracellular organism. And delayed type hypersensitivity reactions carry the greatest risk of all, because the magnification loop is producing some really nasty chemicals, or releasing nasty chemicals and proteolytic enzymes and so on. And they really do cause significant damage to the host. And sadly for Trump, I'll use him again, you know, drinking Lysol certainly will kill the pathogen, but it'll take out the host tissue as well. So it's patently an illogical process to go through. So therefore, what if, and it happens, what if the potential damage outweighs the benefits? You know, if the tissue function is destroyed, there's no benefit to the host whatsoever. And we have evolved some fantastic systems to deal with that possibility. So sometimes, you know, severe delayed type hypersensitivity reactions will not eliminate the pathogen. It becomes chronic, and then we basically form granulomas. And chalazion is our example. You know, if the delayed type hypersensitivity kept on going, we'd lose our lid, and the whole function of the eye would fail. So we basically just wall it off. And we have these enzymes released by macrophages, and the walling off starves it of oxygen, and so we get tissue necrosis, and it also limits the spread of the microbes. So the body at this point said, right, I can't get rid of it, but I'm going to wall it off so it can't do any further damage. And as you know, with chalazion, you can prod them, they're dead, they're not infected. And they'll be quite hard, and quite frankly, so long as they don't make your lid drop down so you can't see, they're actually not causing you any harm at all. So it's a good way of controlling things if the alternative is too aggressive. So again, in some cases, the killing of virally infected cells is just too great to the host. And we have to actually down-regulate things. So for instance, with neuronal cells, they can't regenerate. So if you've got an infected neuronal cell and we destroy the cell, we destroy the function. So obviously we can't do that. And on balance, at least for non-lytic viruses like simplex and zoster, obviously if it's a lytic virus, we've got a different animal because it's about to destroy the cell itself. But for a non-lytic virus, the least harm to the host would be achieved by containing them inside the living neuronal cell rather than killing them. And we know this from simplex and zoster. They lie dormant in the cells, and under normal conditions we keep them well and truly under wraps. But if our immune system is somewhat compromised or we're stressed or whatever, that's when they erupt. So at this point we need to upregulate our barriers and downregulate the immune system. So immune privilege. Right, so what I'm going to talk about now, it's really simplified. I'm not sure everyone understands it. Certainly we don't. I don't understand it. But it is important. It's an important principle. So for instance, the central nervous system, the eye, the placenta, the fetus, the testes, these are immune privileged sites. And they'll tolerate introduction of antigen without eliciting an inflammatory response. So again, back to the example of the neuronal cells, they can't regenerate. And equally, corneal endothelial cells and some retinal cells have limited capacity for regeneration. So again, for non-lytic intracellular pathogens, it is patently not in the host's best interest to destroy the infected cell. Containment is a functional cell is much better. Therefore, neuronal cells, for instance, they don't express class 1 MHCs. So they downregulate the CD8 cytotoxic response to virally infected cells. So within the eye, the anterior and posterior chambers, the vitreous, subretinal space are immune privileged sites. So they downregulate deliberately some of these processes. So of course, when an infection does get into these systems, it's pretty devastating. So after all that, we've sort of come full circle. For these specific organs in our body that have immune privilege, we suddenly have to rely far more on our very, very robust barrier systems for protection. The blood-brain barrier, very highly selective barrier separating circulating blood from the brain, and it restricts passage of pathogens into the brain. Our ocular barriers, we have a blood aqueous barrier, we have a blood retinal barrier, we have tight junctions between endothelial cells and epithelial cells. So the whole system's been down-regulated within the eye. It's an immune privileged site, which incidentally is why you can actually do implants and so on, and they don't reject them because they are, after all, a foreign body. But they don't reject because the whole immune system's been down-regulated. But if the barriers are breached, the impact can obviously be devastating because the immune response isn't actually there. Endophthalmitis being, an absolutely classic example. But so long as we don't do that, we're sitting pretty. So there you are, immunology in 49 slides. I hope that helped. I think it's about 58 minutes long, so hope you enjoyed it, and we'll talk next in the hay fever and allergy chat.